In order to determine the effect of nutrient enrichment on algal cell density, we need to first filter the algae in our cultures. By filtering the cultures, you can see growth variation by how green the filter paper becomes. We will then be able to relate the color of the filter to cell density using a color comparator. This presentation will provide you with the procedures necessary to complete the filtering process. Each group should make sure they have all of the materials needed for the extraction. These materials include distilled water, a dropper, a syringe filter holder, filter paper, a graduated cylinder, a 250 or 450 milliliter beaker, plastic forceps, a color comparator, and your algal cultures. Open the syringe filter holder by unscrewing the top from the bottom. The bottom is the side with the silver mesh screen. The filter chamber will come apart into 3 pieces. In the center of the syringe filter holder is a black rubber O-ring. Make sure not to lose or damage the O-ring, or the filter will not work. Rinse the filter chamber into your beaker with distilled water using your dropper. Rinse both sides thoroughly. Place a filter on the bottom part of the chamber. Make sure the filter is centered and is not damaged. Add 2 to 3 drops of distilled water onto the filter. This will help the filter stick to the bottom of the syringe filter holder and remain centered. Place the O-ring over the filter, making sure it and the filter are centered. Carefully screw on the top of the filter chamber. As you screw the chamber together, keep it level. This will help prevent the filter and the O-ring from moving off center. Fill the filter chamber with distilled water using your dropper. Doing this will help the algae disperse more evenly over the filter and give you a more accurate color. Remove the syringe plunger from the syringe barrel. Attach the syringe barrel to the filter chamber. The plunger should not be in the barrel when attached to the chamber. Measure and add 10 milliliters of algal culture to the syringe barrel. Insert the plunger and push down slowly. You only want to see about 1 drop of liquid per second exiting the filter chamber. When the sample has been completely filtered, carefully unscrew the filter holder and remove the O-ring. Use the filter forceps to carefully remove the filter. Try to grab it at the edge and not touch the green algal coating on the filter surface. Make sure to rinse the filter chamber completely before filtering your next sample. Compare the color of your filter on the color comparator to determine the relative cell density number. Place the filter on your group's filter placement sheet. Make sure to place the filter on the correct circle so you know which treatment it came from. Record the relative cell density number from the color comparator onto the blank line below the circle. When your filter is dry, tape the filter paper onto the sheet and give it to your instructor.